Good morning, everybody. Today we are going to talk about mitosis and the process of cell cycle regulation. And so, really, after today's lecture, you should be able to understand the steps and the structures that are involved in the cell cycle. And then you should also be able to understand how the cell cycle is regulated and then what the consequences are of that regulation being disrupted. Our essential questions are: what are the events of the cell cycle? How does mitosis result in the transmission of chromosomes from one generation to the next? What are the roles of the checkpoints in regulating the cell cycle? And then what are the effects of disruptions to the cell cycle on the cell or organism? And so throughout this lecture, I'm probably going to go pretty fast, so you may need to pause it if you wanted to write certain things down. So just so you know, there won't be a ton of pausing. So why do cells divide? Why do they need to divide? The first is for growth, okay? And so in order to get bigger, for organisms to get bigger, they have to have more cells. And so that makes sense. That's one reason why they would divide. Now, here you can see a picture of my son when he was 1 week old and then when he's 1 month old. And if you kind of see these boxes on this quilt, you can really see how much he has grown in just a matter of weeks. And so really, you never grow more than you do in that, very beginning stages of life. It's just such rapid growth. And so as you can see here, he was growing very, very fast during that time. However, that's not the only reason why cells need to divide. They also need to repair themselves. And so if you ever have a time in which, you scrape your knee or you get a cut on your hand, the cells that are gone, that are, scraped or cut away, are going to be replaced. And so that's another reason why cells need to divide. And then another reason would be tissue regeneration. And so really, as humans, we have different tissues that regenerate. Like your skin is constantly replacing old skin. Your liver is an organ in your body that will regenerate itself if some is cut away. But not too many tissues in humans do that. However, there are a lot of instances in other organisms where if they lose a limb, or if they lose, in this lizard's example, the tail, it will regenerate itself. And then finally, asexual reproduction. And so asexual reproduction is when an organism reproduces without a mate. And so it essentially makes a clone of itself. But that is another reason, or another way in which a cell would need to divide mitotically. Okay, so mitosis is really what we're talking about today. It's the division of the nucleus in cells, but it's really a process where two cells are created that are genetically identical to the parent cell and to each other. And this is kind of the big thing to take away today, is that when we do mitosis, we are making cells that are genetically identical to one another. And so whatever is in this parent cell, whatever DNA is in there, is going to be exactly the same in the two daughter cells that are eventually created. So humans have two main types of cells. And so these cells, are the two different types that we have, are somatic and then gametic. And then, so somatic cells are all of your body cells. Okay, so every cell in your body that's not a gamete is a somatic cell. Gametes are your reproductive cells. And so there are two types of reproductive cells in human beings. Those are sperm and egg cells. And so those are the only two types of cells in humans that are gametes. So obviously females have eggs and males have sperm. Somatic cells are diploid, 2N. So N means chromosome. That's what that symbol means. And so something that's diploid has 2 copies of each chromosome. So that's what 2N is. And then on the flip side, gametes are haploid. They have just a single N, or 1 copy of each chromosome. Somatic cells are always made by mitosis, and then gametic cells are always made by meiosis. And meiosis is something that we will talk about in detail next semester. So mitosis splits up DNA. And so inside of cells, there are kind of 2 different forms of DNA. One is called chromatin. And so chromatin is the loose DNA inside your cells that is not condensed. And so you kind of are thinking of chromatin as this kind of big bowl of spaghetti. And so when your cells are in the chromatin phase, they are loose and kind of all spread about. And then when a cell has chromatids or packaged DNA, it's going to look more like what you would think of DNA looking like in that chromosomal, like condensed form. And so when DNA is going to be condensed, it gets wrapped around these proteins called histones. And so histone proteins are what essentially wind up DNA. There's a really good video of this that I'll show you when I get back to school of how it actually wraps itself. But when a cell is going to divide, it is condensed into chromatids. And so here you can see this is one chromatid. This next picture below it is showing 2 chromatids. So this is 1 chromatid, 2 chromatids, but in both of these cases, this is a single chromosome. So even though there's only 1 chromatid in the first picture and 2 chromatids in the second picture, both of those pictures represent 1 chromosome. This lower picture here is just showing a duplicated chromosome. So it's really important, not as much in this unit, but when we talk about meiosis, that you always count the centromeres when you're counting chromosomes. So every centromere would show you a single chromosome. And so obviously it would be right up here on that single chromosome. Okay, so now let's draw a little picture of the cell cycle. So this right here is going to depict the entire cell cycle. And so if you would just draw this entire picture or this like big circle in your notes, and then we'll kind of add to this picture as we go. So if you need to pause the video to make the picture, I would take a second and pause the video. So this cell cycle is kind of divided into 2 main parts. And the first main part is something we call interphase. So this little arrow circle here is representing interphase. And so interphase is the non-dividing phase of cell division, okay? And if you had a cell in interphase, its DNA would be chromatin. So that loose DNA kind of all throughout the nucleus. Interphase itself can be split into 3 different subphases. And so this first subphase is called gap phase 1 or G1. And so in G1 phase, the cell is growing. It's obtaining nutrients, It's going about, its normal cell functions. But it is doing growing throughout this period. And so after the cell has made it through most of this gap phase 1, this is kind of a crucial point. And so it's either going to keep going through the cell cycle, or it's going to go into a phase that we call G0 or GO phase. And so GO phase is a rest phase for the cell. And this is where it really stops dividing. So there are cells in your body that go into this phase frequently and really don't divide throughout your entire life. And so your neurons are a type of cell that go into GO phase and then don't return back into the normal cell cycle because they don't end up dividing. However, there are, cells in your body like skin cells that are literally going to divide all the time. And so it just depends what type of cell it is, what type of specialization that cell has on whether or not it goes into GO or is in gap 1. So once we have passed through gap 1, we start S phase. And so S phase stands for synthesis. And so in synthesis phase, we are replicating the DNA. So, kind of like that picture I showed you before, if you had a single chromatid, this is the time where it would be duplicating itself into 2 sister chromatids of a chromosome. And so in human cells, you would have, like at this point, 46 chromatids. And by the end, you would end up with 92 chromatids after this entire DNA replication process happens. And so that's really what's happening during the synthesis phase. And then the third and final phase of interphase is gap 2. And we call that G2. And so throughout the gap phase 2, we're going to have more growth and readiness for division. So in this step, the cell is going to be making more organelles so that there's enough in each cell when the cell divides. It's going to grow a little bit more, obtain nutrients that it needs for cell division. But really, it's just more growth that's happening. So then that moves us into the actual cell division phase. So cell division takes place in kind of 2 sections, mitosis and then cytokinesis. We will talk about mitosis first. So mitosis literally is division of the nucleus. So mitosis is when we split up the DNA and the nucleus. And that can be further divided into 4 steps. We have prophase, metaphase, anaphase, and telophase. And so in these steps, different things is happening to the DNA. In prophase, the DNA condenses down. In metaphase, the DNA lines up along the middle plate of the cell. In anaphase, those duplicated chromosomes are being split apart and going to opposite ends of the cell. And then finally, in telophase, we're starting to see 2 new nuclei forming and kind of 2 new cells forming. And then finally, in cytokinesis, this is when the actual cytoplasm splits and you end up with 2 new cells. And so after this, those 2 new cells would just go right into gap phase 1, and then they would kind of start the whole process over again. I think the nice thing about this picture is you can really see kind of the time that cells spend in each part. So if we just kind of like highlight this, you can see that interphase is taking up a lot of the cell's life. Most of the cell's life is going to be spent in interphase, and then a very small section of a cell's life is going to actually be occupied by the division phase. And so a cell, just a little reminder that it does spend most of its life in interphase. And so if we kind of look at the 4 phases, so you can see here prophase is really highlighted by the fact that we can see this chromosomes condensing down. And then as you move into metaphase, you can see all of this DNA lining up in the middle of the cell along the metaphase plate. Anaphase is really marked by the splitting up of this DNA and bringing it towards the opposite ends of the cells. And then finally, telophase, you can really see 2 new cells forming. You can kind of see it starting to pinch off here in the middle. And so then cytokinesis would be when those actual cytoplasms are split into 2. Okay, so now let's move into cell cycle regulation. And so the cell cycle is regulated by a series of checkpoints. And so these checkpoints are really important because they stop the cell cycle if something isn't right. So if at any time one of these checkpoints finds something that's off with the process, it will either stop the process and fix it, or it will stop the process and send the cell into cell death. And so we talked a little bit about this and when we talked about cell communication, but there are protein messengers that push the cells through these checkpoints. And so they're really important proteins to remember because, we're going to talk about them a lot throughout the unit. And so one type of protein is something we call a CDK. And so you can see a picture of that CDK in the bottom right-hand corner here. And so a CDK is something called a cyclin-dependent kinase. It's always present in the cell. So these are proteins that are always in the cytoplasm, kind of in the cell, just waiting around to be triggered by their friends, the cyclins, okay? So cyclins are proteins that are made when they're needed. And so they are made when a cell senses or is messaged that it needs to go through this cell division process. So now we're gonna do just kind of a quick overview of cell cycle control. And so we're gonna talk about just like the main 3 checkpoints that are at these different points in time in the cell cycle. So the first checkpoint, the G1 checkpoint, which you can see right here. So I would just draw a circle, kind of like highlight these main phases, and then you can go ahead and kind of just follow along with me here. But in the G1, phase, this checkpoint really is asking, is the cell good for division? So that's really what's being asked here. And so if the answer is yes, we're going to move into the next phase. If the answer is no, that cell is going to go into G0, okay? Or it's going to be triggered for apoptosis. But either way, The cell is going to have some destination. Now, what, what is this cell checkpoint checking for? Like, what is it making sure? It's asking, is the cell big enough? Does it have enough nutrients? But remember, the signal has to be there, okay? So in order for this checkpoint to get pushed through, there has to be a signal. And so signals that are going to be made in this checkpoint are going to be cyclins, and they're going to be cyclins that trigger in the G1 phase. And so these cyclins are going to meet up with their CDK. And then that complex, that cyclin CDK complex in the G1 phase is going to deactivate a protein called RB, because RB normally blocks DNA replication, which is the next step. But if the cyclins and CDKs deactivate RB, then RB is unable to stop DNA replication, and then the cell can keep pushing through the cell cycle. And so if the answer is yes, then we're going to move into the next phase, which is S phase. And so in S phase, we're just going to have kind of a bunch of cyclins that are triggering DNA replication enzymes. So they're going to activate those enzymes and push them along to really just to make DNA replication happen. And so then the next checkpoint is the G2 checkpoint, which really kind of should be drawn over here. I don't really like where that's drawn. But the G2 checkpoint is kind of checking a couple of things. Well, it's really like if this checkpoint goes through, this is our commitment to mitosis. So if the cell makes it through this checkpoint, like mitosis is going to happen and the nucleus is going to start to get split up. And so it's really checking, is the DNA good? Did DNA replication happen? Are there any errors? And then another thing it's checking is just, is there enough materials for 2 new cells? So if both of those questions are yes, then it's going to push that cell through to the mitosis phase. And so the specific messengers that are going to be triggering this are going to be cyclins that are active in G2 plus their CDKs. Remember that cyclins and CDKs are specific to each other. So specific cyclins only activate specific CDKs. And so that the cyclins and CDKs are going to be called the MPF, or the mitosis promoting factor. And so this is going to activate proteins that break down the nucleus. And then once that happens, once that nucleus is broken down, then that's going to trigger like a chain reaction of events to, kind of make everything else start happening. Like the DNA condensing, the centrioles moving and putting out microtubules and things like that. So next is the M phase checkpoint. And so the M phase checkpoint is really asking, is the DNA lined up correctly? And so if the DNA is all lined up along the metaphase plate correctly, then this checkpoint is going to say, okay, like it's fine, and the DNA can split now. And then the DNA will be allowed to kind of move to the two different sides of the cell. And so the main kind of complex that's activating this here is called the APC, which is the anaphase promoting complex. And so what this is going to do, what this anaphase promoting complex is going to do, is it's going to break down the centromere that's holding the sister chromatids together so that they can split apart and be moved to opposite ends of the cell. And so that's really kind of like the cell cycle control, the checkpoints, and the HHMI activity that you do. We'll go into these in detail as well. And so this just kind of shows you the different cyclins and how they're activated. And it really does make sense. So like obviously if you're in G1 phase, the G1 cyclin is going to be increasingly made, which is going to push it, to continue into the cell cycle. And then this G1S cyclin is going to help. It's going to, obviously be a very high expression at that, brink of the G1S phase. And then as we move into the S phase, it's going to need to have less and less of this cyclin. And then obviously the S phase cyclin is going to get produced a lot during the G2 phase so that it can push it into the mitosis. And then the M cyclin is going to push those chromosomes to be able to be split apart. And so as you can see, the cyclins, like their concentration goes up and then comes back down. So the cyclins are made, they're used, they're degraded, and so then they're gone once, they don't need to be used anymore. So what makes cells want to divide? And what makes cells want to divide is something called growth factors. So these are proteins, they're messenger proteins that are released by cells to stimulate others to divide. And so cells, something important to know about them is they like to be touching, okay? So anytime that cells are not touching, they're going to start sending out these growth factor messages to each other so that they will divide until they are touching each other. And so when they are touching, then they stop sending those signals and they stop growing. Another kind of signal that they send is something called anchorage dependent signals. And so cells only send these signals when they're attached to surfaces. So if a cell is not attached to a surface, it will not send those signals. And so pretty much what that means is you're not going to have a bunch of cells growing on top of each other. Cells want to grow in a single layer. And so they want to be anchored to something, but then they don't want to just be growing in some big, huge mass on top of each other. And so what this kind of looks like is just you would have a growth factor that would initiate a cell communication signal with a receptor protein, and then that would kick off some secondary messenger system. In this case, it's a phosphorylation cascade, and then this is going to activate some protein that will go into the nucleus, and then that's actually going to turn on the making of CDK proteins, and then those CDKs are going to promote cell division. And so if this growth factor was initiated, it would want to activate one of those CDK proteins. So an example of a growth factor would be platelet-derived growth factor. This is a growth factor that initiates the growing of tissue cells. so that they'll grow. Erythropoietin. So this is actually something that cyclists use for blood doping. And so this specific growth factor initiates them to create more red blood cells. And so if they have more red blood cells, they can carry more oxygen, and that extra oxygen goes to their cells and allows them to do more cellular respiration and make more energy, which is obviously going to give them an edge in their competition. And so for a long time, this is something that cyclists would take in order, like kind of like, if another athlete would take a steroid or something like that. And so that's where like the term blood doping comes from. So growth factors in cancer, there's two types of genes that can cause it. You either have a gene that presses on the gas too much, or you have a gene that kind of takes the brakes away. So the first type of gene is something called a proto-oncogene. And so a proto-oncogene, it normally activates cell division. And so you have these genes throughout, your DNA, and they're supposed to be there, and they're supposed to just, make the cell cycle happen when it needs to happen. However, if it's mutated, it becomes something we call an oncogene. And so oncogenes are cancer-causing genes. And so what happens to them is they're always turned on. And if they're always turned on, then they're going to always promote cell division, and that's what causes kind of out of control cell growth and what causes tumors to happen. So kind of the opposite of that is something we call a tumor suppressor gene. And so tumor suppressor genes normally stop cell division. And so those genes, if something's wrong with the cell, they would be like, hey, we need to stop cell division. Well, if a tumor suppressor gene is mutated, it would be switched off. And so if it's switched off, then that would be a cancer-causing gene because there's no brakes. There's nothing stopping the cell cycle. The cells are just going to grow, even if there's something wrong with them. And again, that would lead to a tumor. So in order for cancer to happen, cancer cells kind of have to get over, they have to obtain 6 key mutations. And so one mutation would be unlimited growth. So they're just going to, keep going through the cell cycle. They have either like growth factors that are always on feeding them, or they don't need the growth factor to always be on to signal the growth to happen. They have to ignore cell cycle checkpoints. So cell cycle checkpoints are there to make sure, the DNA is replicated fine and that, the cell is ready to divide and it's healthy. Well, cancer cells have to be able to get around those checkpoints so that they can keep dividing even if they have issues. They have to be able to escape apoptosis. So pretty much that means like they won't die. So most cells only have, so many good divisions in them before they're unhealthy and they just need to go through programmed cell death. But cancer cells avoid this signal. And so instead of going through apoptosis or programmed cell death, they're just going to keep dividing, even though it's not probably healthy for them to be dividing. They have to have unlimited divisions, which is kind of what I just talked about, as many as they want. They're not bound by a certain number. They have to promote blood vessel growth, so they have to be able to get fed. They have to be able to get nutrients and oxygen and water and all the things that they need. So they have to be able to have blood vessels coming to them. And then finally, they got to overcome that density dependence and that anchorage dependence. And so they have to be able to overcome the signals like, hey, all the cells are touching now. We're good. We don't need to grow anymore. And then they also need to overcome that growing on top of each other signal, that anchorage dependence. So cells growing on top and overcrowding other cells. There are 2 types of tumors. One type is a benign tumor. You've probably heard of both these words. So benign tumors, they're still an area of abnormal cell growth. They're not normal cells. They're not supposed to be there. However, they stay in one place, and they're not what we would call malignant. And so malignant tumors, they leave the original site, and they will start more tumors in other places than that original site that they were in. And when that happens, we say that a tumor has metastasized. And so if a tumor has moved on to a new place in the body, that is something we would call metastasizing. Okay, that is it for our notes today. If you have any questions, please let me know.